Hello, everyone. Today we have the great pleasure to have Aadra Bhatt here from the Center for Gastrointestinal Biology and Disease from UNC. And she's going to talk to us about NAMS, which is a subject that is going to hopefully be very, very relevant to many of us very soon if our great grant proposal is going to be funded on the subject matter. So that's why it's a very timely and very interesting subject for us all to learn about. So, without further ado, Audrey, if you would like to share your screen and welcome to the Monarch Seminar Series.
Oh, thank you so much. Really, really appreciate this kind invitation. I'm excited to be here and talk to you all about some of the work that we're doing in my lab studying NAMs. And I'm not sure why here we go. Okay. We're in year 5 of Zoom, and I think sometimes I still forget where all the buttons are. So if people cannot see my full screen, can you please say something?
It's all great.
Okay, great. So all right. I have hidden my video panel just so that I can see a little bit more of my slides in front of me. So please do feel free to unmute yourself if needed, if you have like a really pressing question. Otherwise, as Nico informed me earlier, happy to take as many questions as you like afterward. And my email address is also listed here. Please feel free to drop me a line anytime. So I'm delighted to be here to talk to you about NAMs and how they're at the intersection of bugs, drugs, and guts and rock and roll, and how we're using NAMs to advance precision medicine in my lab. So the way I've formatted this talk is I'm going to give you a very high-level overview of the topic, and then also talk to you with a few specific details about what's going on in many labs around the world and also in mine. And then we'll end with some topics for a discussion either today or another time. So the gut is a really fertile place to study all aspects of biology. It has a very distinct biogeography that is characterized by a number of different gradients. These can be gradients of nutrients, of pH, oxygen, as well as bacterial loads. The gut is the home to many, many diverse microbes, including bacteria, fungi, viruses, and protozoa. And my particular interest is in the bacteria that live within them. And that's because I'm really intrigued by the microbial pharmacists within us. Which is a title of a really fantastic review. I'm only mad that I didn't think of it myself. So the bacteria that reside in our gut are between 100 to 1,000 different species that encode about 1 million genes, and there's 1 trillion different cells within our gut. And these bacteria in our gut are carrying out constantly some very, very complex biochemistry that indeed affects nearly every aspect of our health and also disease states. Honestly, you can't open any popular science book or even the New York Times without hearing something about the microbiome. You go into the supermarket and you see a whole variety of probiotic and prebiotic and postbiotic and symbiotic products that are collectively about $40 billion a year. So the microbiome is really something that people are really excited to learn about. And that's because, again, it's implicated with nearly every aspect of human health and disease, from cancer and infections, obviously, inflammatory bowel diseases, irritable bowel syndrome, inflammation, arthritis, neurological disorders, even like Parkinson's. And what I'm interested in is how microbiota in our gut affect drug responses. So, drug responses are how we respond either positively or negatively to medications. And I became very interested in this field several years ago when I began my postdoc. At this time, there was increasing emerging evidence that bacteria are able to modify the drugs that we take. And conversely, drugs that we take can also modify the bacteria in our gut, not just their composition, but also their function. And this is a very complex field, and I've just sort of cartoonified it here for you to really highlight how medications can alter the microbiota themselves by altering things like pH and osmotic balance, nutrient availability, and in fact, many medications that were not developed to be bactericides are actually having the effect of killing many bacteria. Conversely, in work coming out from my lab and many others around the world, we're also finding that microbiota are altering the way we handle medications, how medicines are absorbed, metabolized, distributed, and excreted, and ultimately whether we respond positively or negatively to them. So there are a number of different factors that contribute to the variability in drug responses. These include things like sex, age, ethnicity, genetics, which are largely not amenable to change, like you can't change those. However, the fact that microbiota also alter drug responses holds a lot of promise for precision medicine because microbiota, unlike age and sex, are actually modifiable. You can modify them, like I said, either with antibiotics or other mechanisms such as the postbiotics or prebiotics or designer fancy sodas and beverages you can buy from the supermarket. So microbiota are modifiable contributors to how we respond to medications, and so hold a lot of promise in improving precision medicine. The way that microbiota interact with medications is a burgeoning field called pharmacomicrobiomics. It's a lot of syllables, and I will throw this word around a little later too, and I'm sure I will trip over it, but it's a lot of syllables, but it's ultimately the study of how bacteria in our gut interact with the medications that we take and how that affects how we respond to medicines. So we're going to get to NAMs really quickly, but I want to give a very brief high-level overview of how medicines are handled when we take them. So here is this purple pill, which is an ibuprofen that I popped earlier today to deal with a bike accident. So when we take these medicines, they are, getting into our gut and they're absorbed in the small intestine. Many medicines that we take they reach the liver for first pass metabolism. They undergo a variety of different biochemical transformations that are carried out by host enzymes. These are enzymes that we encode. These are largely oxidative and conjugative biochemical reactions that then transport the medications to various parts of the body where they can have a therapeutic effect. Ultimately, medications will be eliminated either through the kidneys or through the gut. And when they're eliminated through the gut, that's when they encounter a variety of different bacteria and the enzymes that they encode. And the medications and their metabolites undergo an additional step of biotransformation that we're calling lovingly phase 4 transformation or a phase 4 drug metabolism. And what we're finding is that phase 4 metabolism, or again, modification of drug metabolites by microbiota, is responsible for many of the adverse effects that occur from medications like chemotherapies. Moreover, the fact that microbiomes vary based on people's diet, their environmental status, where they live, geographical region, whether they have a pet dog at home or a pet cat. All of these things contribute to the variability in the composition and function of your microbiome. And so obviously this might be one of the reasons why different people react differently to the same medicine. So ultimately, what my central goal of my research lab is to mechanistically understand how bacterial drug metabolism can explain inter-individual variability in drug response. So what does this have to do with NAMs? Well, the amount of biochemistry that underlies studying how medications interact with microbiota requires a lot of bioanalytical and chemical heavy lifting. And one of the only ways we can really understand what's going on at the drug microbe interface is to develop reductionist systems and new approach methodologies to really get a better understanding of what the heck is going on. So I've organized the remainder of my talk into 3 parts, in which I'm going to tell you how we're breaking down the overall very, very big topic of pharmacomicrobiomics and highlighting 3 different types of approaches or new approach methodologies, which can be leveraged to understand pharmacomicrobiomics and how that can be applied to improve drug response. So part 1 of my talk is to share with you a very brief description of NAMs, or new approach methodologies, to understand the complexity of phase 4 metabolism. So before we delve into this, I just want to make a quick sidebar here. NAMs is kind of a very big, overarching kind of term, and it can encompass everything from, organoid technologies, et cetera, which I think many of us are a little bit more familiar with, but can also include things like consortia of bacteria, or indeed even consortia of stool samples. So the first example here is by my colleague Mohammad Donia's lab at Princeton. So what the Donia lab has developed is called an MDM screen. MDM screen stands for microbial drug metabolism screen. And this is an ex vivo fecal culturing system to map individualized microbial drug metabolism. And the citations are listed here. So what Muhammad's lab did is they collected one donor stool specimen, and they identified the optimal media in which to culture this stool specimen to allow for the maximal recovery of bacterial species. And they were able to use metagenomics to chart the relative abundance of the a microbial population over time, and then also compare it to this PD, that's the pilot donor here. And ultimately, the lab was able to develop a bacterial culture medium that supported the growth of maximal number of the bacterial species that were found in the donor stool specimen. Now, they didn't do this for just one donor. They did this for 20 different donors, again, to capture the variability in people that live around the world, that have microbiomes and eat food. Ultimately, in this graph, I've just showed you again a pilot donor here, and the metagenomics from their fecal sample. And on the right here are the different growth media that supported the bacterial species from this one donor. And you can see that the families that are observed in each of these samples in different culturing media vary, and that's again because of the nutritional requirements of the bacteria and the nutritional components of the media themselves, which support different species. So ultimately, the group was able to identify a pool of sorry. Okay. Sorry, getting a few Zoom messages. The group was able to develop a new approach methodology consisting of 20 human donor stool samples that were cultured in specific bacterial growth media that were then incubated with a number of many medications. So 23 medications incubated with a pool of 20 stool donors. And using very complex LC-MS-based pipelines, the group was able to identify the extent of metabolites that were generated by the bacteria in these stool specimens. So they were able to discover novel drug microbiome interactions to identify metabolites and the specific structures of these metabolites that were generated by gut bacteria. Moreover, they took this work a step further and identified specific enzyme families that were responsible for a variety of these drug transformations. And as sort of the icing on the cake, we're able to colonize germ-free mice with these stool specimens and perform pharmacokinetic modeling and actually recapitulate what they saw in their stool specimens in an in vivo system. So very powerful and beautiful work. Another example of a NAM to study bacterial drug metabolism is from Lisa Meyer's lab in Germany. And in fact, this paper was just published a few weeks ago in Nature. Lisa's lab generated a synthetic community of 21 microbes that were incubated with a number of medications. I realize the font here is really small, but these medications include everything from metformin and amlodipine to classical antibiotics. And using this new approach methodology, the group was able to show the extent of biotransformations of all of these medications that are carried out by bacteria that normally reside in your gut. Finally, the third example of this, and I put simplified in quotation marks because this is a group, Mike Fischbach's group here in California, that generated a complex defined community of gut commensals consisting of up to 119 different species, which can be cultured in vitro and then used to colonize germ-free mice to really understand how in vitro observations can be related to in vivo observations. So a really great way of interrelating what might be happening in a NAM to an in vivo observation. Also, just as an aside, I wanted to highlight the work of my colleague, Simon Gray here at UNC. Simon developed a sort of analogous system in which he was able to colonize an experimental colitis mouse model with humanized inflammatory bowel disease donor stools to generate a reproducible experimental colitis model. And again, we can use the in vivo approach, obviously, or we can actually just study some of the interesting biochemistry that's happening in the standardized bacterial consortium isolated from stools. So this part of the talk was just to kind of describe to you how we can actually expand our understanding of what NAMs are. They can include things like simplified communities or even stool specimens that are standardized and well characterized. So the next step of my talk is to tell you about how we can apply NAMs to understand phase 4 metabolism and how these can be directly applied to improve precision medicine. So I'm going to go a little bit deep into some biochemistry here for a minute, but I promise you it's not too scary. So whenever we take medicines or indeed even some of the things that we make, such as hormones and neurotransmitters, many of these substances are highly hydrophobic. So they're oily, they don't mix with water. And the way our body detoxifies them is in the liver, conjugating these hydrophobic molecules, this big red ball here, with a 6-carbon sugar called glucuronic acid. The addition of this sugar generates what are called phase 2 hydrophilic glucuronic conjugates. These conjugates are miscible with water, and so they're amenable to excretion through the urine or the feces. When phase 2 glucuronic conjugates are excreted through the gut via the feces, they encounter this class of bacterial enzymes called beta-glucuronidases or GUSs. Now, GUSs are really interesting enzymes because they like to chew up glucuronic acid from any glucuronic conjugate. And what happens when this is a hydrophobic cytotoxic drug, such as a cancer drug, this bacterial reaction generates active cancer drugs or aglycones within the gut, which I'm going to tell you has many deleterious consequences. So about 50% of people who are undergoing chemotherapy experience dose-limiting adverse gastrointestinal side effects. And this is typically characterized by diarrhea that's accompanied often by bleeding. And in the case of an anti-cancer drug called irinotecan, this causes very severe grade 4 diarrhea. And sorry. Okay. This is not limited to irinotecan. This is a list of a number of different anti-cancer medications that encompass a variety of different classes of drugs, from aromatase inhibitors to tyrosine kinase inhibitors, each of which is characterized by dose-limiting diarrhea. And each of these drugs is detoxified in our body by phase 2 metabolism and then reactivated by bacterial beta-glucuronidases. So ultimately, this is my why should we care slide. So there are very limited strategies to manage chemotherapy-related diarrhea. In fact, the only real medication that's widely available is loperamide, which only works to a certain extent. There's a hormone that is known to block some of the signaling that leads to diarrhea, but even that's not very well tolerated in cancer patients. So the only way to limit chemotherapy-related diarrhea is to actually take a drug holiday, which is stopping chemotherapy treatment for however long it takes until the diarrhea resolves, which, as you can imagine, has some negative effects, such as your cancer, your tumors growing again. So not a very good place to be, obviously. So this is my reason to do the work that I do and talk about diarrhea as often as I do. So so back to beta-glucuronidases. So the bacterial gusses they're non-essential carbon scavenging enzymes. And this is what a tetramer of the E. coli beta-glucuronidase looks like, the crystal structure. And again, to remind you, beta-glucuronidases will take a glucuronide conjugate and hydrolyze it to generate an aglycone and free glucuronic acid, which bacteria love to munch up and generate energy from it through the Krebs cycle. So what's really interesting about beta-glucuronidase is this is a prototypical phase 4 drug metabolizing enzyme that, in work that my lab has done over the last several years, and others around the world, we have demonstrated using a variety of different approaches, both in vivo and in vitro, that this is the enzyme that drives drug toxicity. And what's really helpful is that you can actually measure the rate at which this bacterial enzyme operates using really fancy bioanalytical techniques. Moreover, in work that we've done over the last several years, we've identified specific selective and non-lethal inhibitors of beta-glucuronidases that can block this reactivation activity in vitro and in vivo models. Importantly, blocking beta-glucuronidase activity does not actually affect the anti-cancer activity of cancer drugs. So again, we're really hopeful that drugging this beta-glucuronidase axis is a way to help alleviate some of the chemotherapy-related diarrhea observed in people. So through some data mining that we did a few years ago, we found that pretty much every member of gut bacterial families encodes a specific type of beta-glucuronidase enzyme. And moreover, we've done a lot of important structural elucidation of these enzymes and identified structural features that are responsible for reactivating drug glucuronide conjugates of various flavors. Furthermore, we've developed a number of specific selective inhibitors to beta-glucuronidases to target this bacterial turnover activity. Now, here's another why should we care slide. So in mining the 139 fecal databases in the Human Microbiome Project, we found that about 2/3 of individuals encode bacterial beta-glucuronidases that are known to be drug reactivating enzymes. And again, this is work that we've done in the lab over the last several years to identify the specific molecular features that drive a drug glucuronide reactivation. And I've listed some of the citations here. So that's well and good about chemotherapies, but what about other medications? In fact, lots of other medicines, including pain relievers, medicines to manage cholesterol, diabetes, psychotropic medicines, hypertension, and also illicit drugs like psilocybin and THC are all eliminated by glucuronidation. So ultimately, these beta-glucuronidase enzymes are really important for how we handle medicines. And in the interest of time, I just want to very quickly tell you a small anecdote, which is about inflammatory bowel diseases, so Crohn's and colitis. So people who have IBD have a flare of their disease that occurs due to things like their diet or stress, smoking, acute infections, of course. But NSAIDs, non-steroidal anti-inflammatory drugs, are known to cause flares in IBD. So not only is pain management a really big problem for those with IBD, there's also a pretty high prevalence of narcotics addiction, because again, these are the only way that folks can handle their pain. So there's been a critical need to develop new therapeutic approaches to manage IBD-related pain. And this was the work that sort of actually got me started on pharmacomicrobiomics in my independent lab. So my lab mined the IBD metaomics database, which is a database containing metatranscriptomic, metagenomic, metabolomic sequences of individuals who have IBD. And we actually found a pretty high number of drug reactivating enzymes in individuals who have IBD compared to healthy donors. And we used LC-MS, MS approaches to actually quantify the rate of reactivation of NSAID glucuronides in individuals who have IBD versus healthy controls that were aged and sex matched. And the reason we did this is because NSAIDs have been around forever and their pharmacology is very well described. It's known that a higher rate of reactivation and a higher amount of local NSAID concentration is known to cause more topical injury. And so using bioanalytical techniques, we actually demonstrated that individuals with IBD have a much higher rate of turnover of NSAID glucuronides in vitro compared to your non-IBD controls. So this brings me to present to you another NAM, or proposed NAM, which is stool specimens in a method that we're developing in my lab called in-femo reaction phenotyping. So we can quantify the rate at which the reactivation of a glucuronide conjugate turns over into its aglycone. And we can convert this turnover rate as an input into physiology-based pharmacokinetic models and relate that to the amount of enteropathies that are observed in people taking chronic dosage of NSAIDs, for example. So the word in-femo is an expression that I developed with the help of a classics professor, Luca Grillo. Luca tells me that in-femo is a word that the poet Virgil would have used to describe feces. And I propose to you this is a polite way of talking about stool specimens in mixed company. So in-femo reaction phenotyping is a method wherein lysates are prepared from a given stool sample, and they're incubated with either a fluorescent reporter substrate to quantify the rate of reaction, or an authentic glucuronide substrate. And then again, that turnover is quantified using a slightly more involved bioanalytical assay. And what we're doing in my lab right now is quantifying this turnover and using that rate as a way to predict who is going to have a higher GI side effect or a worse GI side effect in response to a given medication. This could be from a cancer drug or something like an NSAID for individuals who have IBD. So, so we think that in vivo reaction phenotyping is a way to introduce precision medicine to a new NAM, which is again based in biochemistry and biology. So leave you with this little plug in Fimo Veritas, stool as a NAM to predict drug responses. Okay, now I'm gonna, for the last part of my talk, I'm gonna actually tell you how we can leverage NAMs to predict adverse effects of phase 4 metabolism. So this is where I'm gonna tell you about prototypic NAMs. So these are NAMs that new approach methodologies that I think maybe people are a little bit more familiar with, which is organoids. And again, I'm going to focus my talk very much on pharmacomicrobiomics. So I'm going to specifically talk about NAMs that are related to the gut. So what are organoids? These are structures that are derived either from stem cells or progenitor cells. These can be either embryonic, where legally and ethically possible, induced pluripotent stem cells, or even adult tissue, which is what we do in my lab. These organoids can be grown either two-dimensionally as flat monolayers or three-dimensionally in a jelly-like substance called matrigel. Organoids exhibit self-organization. They mimic organ architecture at a very tiny scale. They contain usually all the relevant cell types that are specific to the tissue of origin. And importantly, they recapitulate organ-specific functions and have the requisite gene expression programs that enable their differentiation into the different cell types. So NAMs, organoid NAMs are really great models for studying development, for studying physiology, for studying diseases, or even treatments for diseases, regenerative medicine, and drug testing, which is what we do in my lab. So all of these organoids have very variable provenance. As I alluded to earlier, they have different types of starting material. So some gut NAMs or gut organoids are derived from biopsies from such as those collected during a routine colonoscopy. In someone who's having a screening colonoscopy. These could be surgical resections, such as those undergoing bariatric surgery. Again, this is something that we use a lot in my lab. Organoids can also be developed from pluripotent stem cells, fetal tissues, and also, importantly, organ donors and biobanks. So in North Carolina, we are home to HonorBridge, which is the largest organ procurement organization. They're just kind of down the street from us here at UNC. HonorBridge recovers organs for transplantation, obviously in those who need them, but then also provides tissues for researchers like my lab. So we've had a very productive collaboration with them over the last several years. In 2024, HonorBridge was able to connect with 350 organ donors, 1,372 tissue donors. In my lab, our tissue bank from HonorBridge is in the high teens at this point. So we're very lucky to have multiple ways to access starting material to develop NAMs to study drug microbe interactions. Something that's new and developing, and largely due to my friend and colleague Heather McCauley joining our group, is the increasing usage of iPSCs, iPSC-derived organoids. So Heather brings with her technology to cultivate human intestinal organoids, both in cell culture, as well as by subsequent implanting into the kidney capsule of immunodeficient mice. So when engrafted into the kidney capsule, these human intestinal organoids actually mature and develop three-dimensional structures that are very reminiscent of what the gut looks like in people, both adults and in children. So this is a new approach methodology to study drug-microbe interactions that we're very lucky to have here at UNC. It's always fun to see some of these pictures. Here's a representative picture of an ileal organoid that was grown in my lab. You can see how the organoids grow over time. Within a week, you have this large ball of cells, which can, when examined later, up to 3 weeks later, has actually formed the crypt villi structures that are, again, reminiscent of what goes on in your gut. And the way I encourage people to think about this, who don't think about the gut all the time, is, the gut is basically a tube, and the hole in the middle is where all the business that we're interested in is happening. And that's home to the bacteria, as well as fecal effluent, medications, things like that. So you can think of the inside of this organoid being the inside of that tube. So although this particular model is largely inaccessible for us in 3 dimensions, because again, we're interested in what's happening on the inside of this ball of cells, it's still a really great way to have a spatial understanding of how infectious agents, for example, interact with the gut. So just as a sidebar, my lab is currently working on developing what are called apical out organoids, in which we can actually grow this organoid inside out by just switching out the growth factor that these cells are cultured in. So stay tuned in the next year, hopefully. But what is very interesting about these 3-dimensional organoids is you can basically disassociate them and culture them on transwell or porous membranes and encourage this 3-dimensional structure to now grow as a 2-dimensional monolayer. And what this now allows is for us to access this inside of this tube in what's called the apical compartment of this monolayer growth system. And again, by fine-tuning the growth factors these cells are cultured in, we can now revert this 2D monolayer to now also go back into its 3-dimensional orientation. So some really cool biology that we can do here to really help us answer our biological question du jour. So again, my interest is in studying phase 4 drug metabolites. So for us, the monolayer system grown in 2 dimension is the just sort of the perfect thing for us. When we look at a cross section of the cells grown in a monolayer, we actually see appropriate organization of cells that that reflect the same apical basal patterning that is observed in the gut. When you look at the cells on FOSS, so as if you were looking down on them, you can actually see a highly proliferative layer of cells that are at the edges, whereas more differentiated cells that lack mitotic capacity towards the center of these monolayers. Differentiated cells express all the various tight junction proteins that are, again, characteristic of the gut that helps to create a tight seal, keeping, the rest of the world outside us and, from the rest of our periphery. So this is a great ideal NAM with which to study phase 4 drug metabolism, among a lot of other things. I'm going to just leave you some snippets of what we can do and why we need help interrelating lab observations to what actually happens in people. So a typical workflow in my lab is to collect these cells from donors, culture them as organoids, and then sort of flatten them out into monolayers, and then perform high, medium, or low throughput studies with which we can study various aspects of cell physiology. So these are things that could be insults in response to different phase 4 drug metabolites and conjugates. So these can be things like measuring cell viability or the generation of reactive oxygen species. We can quantify barrier defects, so the tightness with which the guts that are grown as 2D cultures are able to exclude the environment from the rest of the cell system. And then we can also do slightly more involved and laborious, but highly informative studies, such as quantifying cell swelling and calcium signal. So as an example, our NAMs derive from the ileal monolayers of human small intestine, and cultured as stem cells are highly proliferative. So red means the cells are happy and growing. But when we incubate these cells with physiologically relevant concentrations of a cancer drug that is reactivated by gut bacteria, we now actually see this red signal indicative of proliferative happy cells disappear in a dose-dependent manner. So these concentrations used are reflective of what's actually found in people undergoing chemotherapy with irinotecan, which is a drug used for colorectal cancer and pancreatic cancer. So we're able to really model drug toxicity events in the stem cell pool. Furthermore, we can incubate our differentiated cell monolayers with increasing concentrations of diclofenac, which is a non-steroidal anti-inflammatory drug used for chronic arthritis, or your occasional bike accident. And you can quantify the amount of superoxide that's generated in these cell monolayers. And again, that's proportional to the amount of cell death that's observed. You can also see that similar physiologically relevant concentrations of diclofenac cause breakdown of the epithelial cell barrier. So they cause barrier defects, which again is colloquially called leaky gut elsewhere. The same model system can be used to quantify microbial virulence factors and ways to inhibit them. This is work that we've done in my lab with a protozoal parasite. But then finally, I want to leave with this last data slide, which is when we take the same concentration of a phase 4 metabolite and incubate it with cells derived from 4 distinct human donors, we actually see a totally different response to this one specific phenotype, which is barrier leakage, quantified by this fluorescent reporter. We see that donor 1 is very highly leaky in response to many of these medications that are used, some of which are actually some that you wouldn't even think would be causing barrier defects, such as metformin, whereas the others are largely resistant and don't seem to be highly impermeant to these medications. So I'm ending with this slide here to kind of highlight how, again, we sought to develop all these different tools to understand variability in drug response, and we ascribed it to the variability in how microbiota metabolize drugs. But at the end of the day, we find that even our NAMs that we're using to quantify phase 4 metabolites itself have a bunch of different variability. So I'm going to leave here with telling you about the advantages of organoids and other new approach methodologies and how they can be leveraged to enhance precision medicine. So they have reduced complexity, so you can quickly elucidate mechanisms. You can do experiments on a larger scale, which would not be feasible with animal models or even with people, obviously. You can decrease cost overall. But then obviously there's limitations. These are still artificial systems. There's technical limitations such as the inaccessibility of the lumen that I told you about a little earlier. And this is still actually quite expensive. The growth factors that are required to culture some of these organoids, or the amount of regulatory burden we incur to collect stool banks, for instance, is also rather still expensive. So it's not like $1.57 hot dog or anything, you know? So um, ultimately, the open questions here that I encourage, discussion with my lab, with you all. So what makes a representative NAM for a given phenotype, right? Um, Again, in my instance, that's drug metabolism by microbiota. How do we control for the variation that we're seeing? How do we determine a sample size? How many experiments do we actually have to do to capture what is going on in people? Can we even do that? How do we interrelate the phenotypes, such as chemotherapy-related diarrhea, with cell-based assays? So these are things that my lab plans to work on for the next however long it takes. So finally, the key takeaways here are bacterial drug metabolism has an underappreciated role in driving drug responses. Microbiota are, yes, they're super interesting, but they're important because they're modifiable drivers of drug response. And the microbiome space is ideal to incorporate and indeed embrace new approach methodologies to advance precision medicine for all. So I've been working on this with a small team for several years, but my team has just rapidly expanded in the last few months. Very grateful to everybody's hard work. We're so reliant on core labs here at UNC, and also collaborators that have been very generous as I started my independent research career somewhat recently, and obviously my funding sources. I've thrown a lot at you all in a relatively short amount of time, so I anticipate questions. I'm very open to them, so fire away.
First of all, Aadra, thank you very, very much. There were a lot of very excited back channel conversations with people that you won't see directly here in the chat, with like fans of this work. So it was really great to hear. And I, for one, will definitely never forget this idea that I would take a pill, like maybe I would get treatment for some disease, and it might be that the constitution of my gut microbiome will actually affect the effect that this drug has on me and the treatment that I receive. So this concept alone is just like, for me, very interesting that I learned it, and all this what you said about the NAMs was very far beyond me personally. But I will listen to this talk 3 times again so that I understand it much better.
Oh, thank you.
And
You're so
thank you very
sweet.
much.
Thanks.
All
Oh,
right.
yeah. Yeah, thank you.
Uh, no, no, that was great. So yeah, please, everyone here, fire up if you have any questions. Yeah, I don't know whether maybe Harry, you want to get started here.
Sure. Yeah, and I just want to echo Nico's thoughts that this is really exciting stuff
Oh,
presented
thank
very
you.
very clearly. So this is very very exciting. And one thing that I did want to highlight that I was really glad that you touched upon was the fact that the microbiome is something that we have some kind of influence over. So it does make sense that this would
Yeah.
be an
And
ideal. Yeah.
academically, if you will allow me to interrupt you, and academically, this is again a very fertile ground because the microbiome evolution has occurred in like the era of super cheap sequencing and big data. So like there is so much data. There are bajillion, what's bigger than a terabyte? I don't even know, but there are terabytes of data out there that are open to, being leveraged to actually improve people's health. So these are things that an experimental biologist like me can only fantasize about, So I think this is a really great space to kind of like move forward into using machine learning and, you know, other approaches that I wouldn't even know where to start with. Yeah. So.
So I did have one question, and I'll preface this by saying that I've been talking a lot lately with people interested in NAMs from the perspective of modeling adverse outcome pathways. So they're very interested in it. You know, I mean, the basic biology is great, but what they really want to know is, okay, given this type of exposure, what's going to happen? What you know, or in many of their cases, is this something we need to care about from like a regulatory perspective? So I guess my question here is, so since the organoid based platforms are still relatively new and relatively expensive, and but potentially very informative, what do you see as kind of like the um immediate areas of progress for gastrointestinal organoids, specifically beyond the work that you're doing right now with kind of the drug sensitivity?
Yeah. So there are, I think right now, we're all really, I'll speak for me, maybe. I guess I won't speak for all other experimental biologists, but like, I'm really excited by the fact that we can model responses in a dish and personalize who's going to react well to a medication or not. So I'll give you an example. Let's maybe switching away from organoids, let's talk about poop for a minute, because I hear there are lots of poop fans who like to talk about poop on this call. So I'll give you an example of a medicine called that's used for ulcerative colitis. And this medicine doesn't actually work in a lot of people. It like works in a very few people. And in fact, they have to take gram equivalents of this medicine called sulfasalazine for it to have any effect. And we recently discovered that that's because there has to be a very specific microbial enzyme in your gut that can actually convert that medicine that happens to be a prodrug into the active drug. So unless you actually know that the individual you're prescribing this medicine for actually has bacteria that express those enzymes, it makes no sense to give them that medicine, right? So you're just kind of unfortunately, you're forcing them to live with their ulcerative colitis and take medicines that have absolutely no effect. So I see these NAMs as a way to actually, you know, potentially, I mean, bear with me here, but a point of care type of tool where you can take a stool, use a dipstick type device in which you can actually quantify whether that prodrug conversion is happening, you know, while you're in clinic getting assessed by your gastroenterologist. And if that dipstick turns red or, you know, whatever, then guess what? You're actually going to be a really good candidate for this medicine. Whereas someone else might benefit from a biologic to control their UC. So that's one, you know, kind of pie in the sky vision that that's sort of related to what I talked about today, which is, well, if you have, this is during drug development, okay? So drug companies don't actually integrate the role of the microbiome in, you know, some of the adverse effects that arise from their medicines. These things don't actually come up until your human trials, right? And at that time, sometimes medicines have to be shelved because of adverse events. And so if you were actually able to develop tools that can quantify or, you know, even identify ways in which drug metabolites are generated of varying, you know, flavors by your intestinal microbiota, we are presenting a gut tool, like a NAM, that can quantify the different types of cytotoxicity that could occur. I mean, barrier permeability is just one example. You could just have a severe diminishment of the stem cell pool, such as what we've seen with irinotecan metabolites. So this is a way to, by integrating the role of bacterial drug metabolism and then examining that using organoid technologies, you can actually maybe do some med chem and modify the chemistry of the molecule such that rather than it being subject to glucuronidation or sulfation, it's actually glutathione related, you know, and so the enzymes that are glutathionolases are not that prevalent in people as such. So that's one possibility. Sorry, very long answer.
Oh,
Yeah.
no, that's yeah, fantastic examples. Thanks.
Okay, is there anything in the chat? Well, I see a question from Sarah. I feel from our monarch side of things, this fourth question of how to interrelate phenotypes with cell-based is interesting to think about. Yeah, we, you know, open to future discussions at any time, Sarah and others. So, well, if there's
Maybe I'll just kind of jump in and say that was fantastic talk, Aadra, as well. So
thank you, Chris. Yeah, thank you.
I probably have like a million biological questions, but don't have time for them. But I just wanted to let people on the call know that we have been developing our new approach methodology ontology, and it was very inspired by earlier talks with you and so on. And even as you were giving your talk, I'm having kind of ChatGPT transcribe some of your pictures to see how we would model the data in NAMO, if that's okay with you.
Yeah, sure.
Yeah.
Yeah. I mean, that's again, that for an experimental biologist like me, stuff like this is the dream, because if we can miniaturize or automate or, you know, AIify some of these things, ultimately to like reduce the burden of chemotherapy related diarrhea, like that's a really big problem because this is a quality of life issue. You know, you're already very sick and your radius is already limited. Now your radius is limited to your toilet. Like that's just not the way anyone should have to live. So yeah. Anyway, well, thank you all for your kind invitation. I really appreciate it. I really love the enthusiasm. Please get in touch if you would like to talk, and, you know, develop some of these things offline together as a group. Very open. I'm a relatively new investigator, and I'm deciding to dream big right now. So yeah.
Thank you very much, Aadra, for
Yeah.
everything. And
Well,
yeah, you will definitely hear from us.
all right. Well, thank
All
y'all
right.
very much.
Let's thank
Have
you
a
again
wonderful
and
day.
have a great
Yeah.
day.
Yeah.
Bye.
Take care. Bye.